Hey, it's Miss Hunt. This is Tuesday, the 17th, and this is for your homework for today. First of all, if you were absent today, we finished up going over our notes from yesterday, which was the cell cycle division notes. For you all, I've recorded it already on the Screencastify. Something else that my kids from my other classes were struggling with, just the basics of mitosis, the cell cycle, kind of confused on all of it. So I made a document here called Lost Students, and I made a video in here, and I've put some other pictures of some things that might help you if you're just confused on, just the basics of DNA synthesis, protein synthesis, and the cell cycle. So if that video helps you or the documents help you, then that's great. All right, today in class, we did the cell cycle POGL. We didn't finish it. We have to finish that last little bit, which is important because it has apoptosis on it, which is something we haven't covered enough. So I want to make sure that you guys do finish that POGL. I am taking that for a grade, so make sure if you're absent, you're turning that in. Tomorrow, we have another POGL. So make sure in this document, I've got a bajillion things on here. So make sure you do view more so you can see all the stuff, okay? But I wanted to go through the cyclin cell cycle regulation notes. I need to go through that with you today so that we can do the cell cycle regulation POGL tomorrow. So this is part of your homework, is to watch this video and see if you have any questions about the control of the cell cycle. And if you want to finish up or can finish up the POGL from today and go through apoptosis, then that would be great. Okay, so I think it's important for us to remember that this whole unit is about cell communication and the cell cycle. So we want to be thinking about that the cells are communicating through all of this. Even through the cell cycle, there's communication that's happening. So we went over this today just to finish up the notes, and this is also from the previous slide, that cells are going to go through these checkpoints. And then we talked a little bit about cancer. It's going to go a little bit more into this about cancer. All right, the frequency on which cells divide, it determines is by what kind of cell they are. So embryonic cells, those reproduce super fast. And think about it, you're going from one cell into a baby, and from a egg to a blastula to a embryo to a fetus to a baby. All this happens in 9 months. That's crazy how fast it happens. So the embryonic cells reproduce super quick, less than 20 minutes. Skin cells on average are 12 to 24 hours. Your liver cells stay in G0. So remember, G0 is like where they're not going into S phase. They stay there, but they have the ability to turn on and to make a new cell. So they will go into the S phase when they need to. And about every 2 years, your liver turns over a new set of cells. Mature nerve cells and muscle cells don't divide after maturing. Sometimes they stay permanently in G0 phase, where they're not going into the S phase, especially our nerve cells. Muscle cells, maybe after maturing. That's why it's important for us to build muscle on when we're younger, so that when we get older, we are pulling from that muscle, or at least maintaining that muscle. Our immune system, periods of slow division followed by a burst of rapid division upon an infection. So we don't always just keep our white blood cells ready to fight off an infection. And the red blood cells, I wish I could catch that a little bit better here. The red blood cells, maybe I can hit hide. There we go. 2.5 million die and replaced each second. There's another one here. Okay, I'm going to exit there because you just can't see it. There we go. I probably messed it all up now. There we go. Okay. 2.5 million die and replaced each second. Your whole blood gets replaced about every 120 days, which I found that so fascinating to think about. A red blood cell, when it bursts or when it does apoptosis, when it falls apart, it implodes inside, but the membrane of it stays in your bloodstream as little platelets, and that helps form scabs. So those platelets are really important to heal from a surgery or an injury, and it are part of your scab. And they're replaced every 120 days. Okay, so as we go through the cell cycle, your body will stop and have little checkpoints to make sure you're doing everything correctly. How amazing is that? That your body stops and proofreads and checks. So here we go through interphase, G1, S, and G2. We go through G1 and we stop and we have a checkpoint to make sure that everything is going correctly. If it passes, we go to the S phase. We go to G2. We stop at G2 to make sure that everything was synthesized correctly and we're prepared to go through mitosis. Then we go through mitosis and we go to the M phase checkpoint before we finally make our new cell. Okay? The control system of a cell operates by a series of checkpoints found during G1 and the end of G2 and the M. Okay, they're stop and go signals. And they take inventory. The G1 restriction point is the most important one. So this one right here. If you are a no-go, then the cell can either do apoptosis or fix the things, but that's basically stopping it from coming through. Most cells are in the human body are in the G0 phase. Remember, from here, we can loop out and do the G0, where we're just kind of in limbo. We're not going to the S. Our brain cells, our spinal cells stay in G0. They're not going to make new cells. So if we get a brain damage or a spinal injury, it's not going to heal back and come back and make new cells. Okay. Major checkpoints of this cell cycle. Can't get my picture there right. All right, the G1 and S right here. Restriction points. Will cell division occur or not? The G2 to the M, so G2 right here to the M. So here was that first one right here, I'm sorry, the G1 to the S. Okay, and then here is the G2 going into the M, and then has the DNA replicated correctly. Then we have a spindle fiber checkpoint. If everything's correct, the sister chromatids separate correctly, and then we can go on. Okay, so even with all of our POGIL from today, the kids were like, this stuff is pretty easy so far. So, and for the most part, this has been pretty easy. There are numerous control molecules involved in the cell cycle. So we're going to use two of these control molecules. We're going to talk about cyclin and cyclin-dependent kinase. They label a cyclin-dependent kinase as CDK. Okay. All right, kinase, an enzyme that is involved with phosphorylation. So you should be thinking everything's going to be passed down from one thing to the next. It activates or inactivates other molecules, a transduction, that's the word I'm looking for. We're going to see a transduction happening here. Kinase drives the cell cycle and are present in constant amounts. And most of the time they're inactive, and cyclin activates them, hence the name cyclin-dependent kinase. So the cyclin activates the kinase. Okay, and there are many types of cyclin-dependent kinase activated by many cyclins. So here you see CDK cyclin is activated with CDK1, which activates cyclin A, which activates CDK2. So we're seeing as a transduction here of passing it down all the way through. Same thing for a phosphorylation. Everything gets passed through from one to the next to the next. Okay, this is the cyclin, and it's binding to that CDK, and here's the phosphates that pop off for a phosphorylation. So this is the mitosis promoting factor. It's also called the MPF. Okay, cyclin is a group of proteins that binds to the CDK and activates it. Binds to the CDK and activates it. Unlike CDK, it is not there in a constant amount and cycles throughout the cell cycle. When CDK and cyclin combine, they form the maturation promoting factor, the MPF, the maturation promoting factor. The maturation promoting factor activity is directly correlated to peak cyclin activities. So we see here's the MPF cyclin and then the MPF CDK. So we see them at the maximum peaks coming up through each one of these. Okay, to me, like this was like a transduction pathway. So the cyclin binds to the CDK, and then we go to the next phase. Kinase operates here. We have the phosphorylation popping off. Here's our phosphate popping off, and we're activating and going to the next thing to trigger that target protein to do what we want it to do, which is to synthesize DNA or go into the next phase of the cell cycle. Okay. The maturation promoting factor is in charge of phosphorylating many molecules and creating a plethora of cell signal changes. That's a good word, plethora. It means a lot of a variety. Fragmentation of nuclear envelope, chromosome condensation, spindle fiber formation, and cycling destruction at anaphase. So all these things are happening because of that. And then we got another graphic here to look at. Tomorrow when we do our POGL for regulation, you're going to see a lot of the checkpoints, and we're going to talk about that. You also see some graphs like this in the POGL. So I think we're going to see this again and be able to understand what we're talking about. All right. There are negative regulations of the cell cycle. A negative regulation ensures that the cell growth does not go uninhibited. So this is going to stop the cell growth. So I had on here that we were going to do negative and positive feedback loops, but I didn't get that together. So in a negative feedback loop, we have inhibitions. So a negative feedback causes you to go back to your original spot, which is good because that's what you want, is to go back to your original spot. So our cells make a cell cycle and we go back to our original spot. We go right into the G1 phase again. So we're just continuing in the same pattern. So as a negative causes you to go back to where you came from. A positive feedback loop continues all the way down and you move farther away from where you originally started. So they kind of have a misnomer. Negative sounds bad and positive sounds good, but they're not always that way. Positives usually cause the most change in the body or in anything, in any system. And negative causes it to go back into its original spot. Okay. All right. There are many classes of these molecules, but the most common is the retinoblastoma protein. Okay. This protein are tumor suppressor proteins, and they halt the cell cycle if it's the DNA is damaged. It can signal apoptosis, which is the destruction. These things right here are just different types of proteins that can bind into that maturation promoting factor. Okay, or the MFP is the something different. Nope, that's the MPF. I'm thinking maybe that's a typo, that it should be the MPF, okay, combined to inhibit the maturation promoting factor in the cell. All three types operate primarily at the G1 checkpoint. So that's why the G1 checkpoint is so important, because all of these things are being checked off there. Okay, the RB monitors the cell size. The RB, when not active, binds to the transcription factors, turning them off so they cannot bind to DNA. So remember, the transcription is when we are going to make the mRNA. As the cell grows, the RB slowly becomes inactive and thus activating the transcription factor that it's bound to. For the cell to move past each checkpoint, all positive regulations must be turned on and all negative regulators must be turned off. So every promotion needs to be turned on to go through the next part of the cell cycle. So this is that positive control. So this is moving farther away from the original, okay? And we're promoting from one to the next in a positive feedback loop where we keep moving down and our DNA gets synthesized, okay? External control of cell growth. Cell growth factors are chemicals that are released by cells that stimulate the cell division. Growth factors bind to receptors inhibiting signal transduction pathways that encourage cell division. There are at least 50 different growth factors that stimulate cell division. So these are our little growth factors that are promoting this. This is an epidermal growth factor. So this is on your skin. Okay. All right, there are things that are going to inhibit or stop growth. There is density-dependent inhibition. Crowded cells will stop dividing when there's not enough food or resources to continue growing at that rate. So each cell binds a bit of its growth factors and stops it. If there's not enough activators left to trigger the cell division in any one cell. Anchorage dependence to divide cells must be able to attach to a substrate, so like a touch sensor. So here in the little flasks, normal cells are here, but if we deprive it and take away any nutrients, there's nothing for it to divide. This is cancerous cells will continue dividing even on top of each other without the promotion. So you can see like they should have realized there's no more resources, but they didn't. And that's why they're just continuing on. Okay. In order to get a cancerous tumor, or get cancer, you have to go through the cell cycle and hit 6 key mutations. So I want you to remember, cancer is still really rare. Although we all know somebody who has been affected by cancer, cancer as a whole is a rare thing. Unlimited growth. Your growth promoter has to be turned on so that you can keep growing and growing and growing more and more cells. You have to ignore all checkpoints. All of them have to be turned off for tumor suppressor gene P53 has to be turned off. You have the ability to escape apoptosis. Apoptosis is programmed death when you've gone wrong. So it has to be able to turn off this suicide gene to kill itself and stop it. That can't be working properly. Immortality. You have to have unlimited divisions where it just keeps going. So the maintenance gene has to be turned on. You have to promote blood vessel growth. So all this has to be turned on and overcome anchor density dependent. So that has to be turned off so that you can keep going and growing even if you don't have enough nutrients. So that lets you know that in order to get a cancerous tumor, you have to be able to get all of these mutations right or wrong in order to get that cancer. Okay. All right, so this is the positive regulations. Remember, they're moving forward down a pathway, and these are the negative that return you back to your original, okay? All right, what causes the hits? These are any kind of things that can trigger a mutation. UV radiation, a chemical exposure, radiation exposure, heat, cigarette smoke, all these things. And there's so many other things that are known carcinogens that can cause cancer. This is a breast tissue, and you see the glands that are inside the breast, and you see a tumor that's formed of just one cell. That tumor is growing and spreading. And then here's the lymph nodes, and your lymphatic system runs throughout your whole body as a bunch of lymph nodes. So if it invades into that lymph nodes, then it becomes a metastasized or a metastasis, and it's spreading throughout the body. Okay, something else we talked about in class today, if a tumor is benign or malignant, a benign tumor is abnormal cells remain at the original site as a lump. The P53 has halted cell division, and most do not cause serious problems and can be removed by surgery. So remember also we talked about a benign tumor, also where it happens is a problem too. If it happens in the brain or lungs or heart, it can become a problem. But in just the breast or on your arm, it's just a lump. And if it's not doing anything, it's okay. Malignant is when it's a dangerous situation. The cells leave the original site, they lose attachment to nearby cells, and they're carried by the bloodstream or the lymphatic system to other tissues. And they start more tumors throughout the body. It metastasizes and spreads throughout the body. Impaired functions of organs throughout the body can happen with a malignant tumor. Okay, that's it for today. Okay, we didn't have much. Exit here. Okay, so we didn't have much to do, which is good. I'm trying to find how to turn my recording off here. Okay, there we go. All right, so what do you got to do? After you listen to my notes, finish your POGIL from today, the cell cycle. If you didn't do your Bozeman yesterday, do your Bozeman. Tomorrow, we got a whole bunch to get done, and hopefully you go through these notes so you're ahead or you're on schedule. All right, great. Thanks.